was Jennifer Doudna. So she was born in Washington, D.C. and moved to Hilo, Hawaii when she was only 7 years old. Her father taught English literature at the University of Hawaii, and her mother taught history at the local community college. When she was young, she always felt out of place because she was from a very white background and could not relate to her fellow classmates that were of Polynesian descent. And her parents were also from a background of education, which was just not very common where she was from. So, because of this, she read a lot of books and about the nature and the landscape of Hawaii. Her passion for nature quickly turned into her passion for science. During one summer, her parents sent her to spend time with Don Hermes, a biologist at the University of Hawaii who's also their close family friend. During her time with Hermes, she learned how calcium ions play a role in the development of fungus. Her passion for biochemistry especially started at the age from 12 to 13. This really sparked when reading about James Watson's discovery of the double helix and after hearing about a female scientist speak about how cells can become cancerous. So, for her bachelor's degree, she obtained a degree in chemistry at Pomona College, and this was during 1985. She chose this college because it was small and she felt like she could get a good education. During her time here, she worked in Sharon Panasenko's lab, and she really admired her because she was a very successful female in a very male dominated field at the time. And for her doctorate, she went to Harvard University, and this was in biochemistry during 1989. And she worked under the supervision of 2009 Nobel Peace winner Jack Szostak. Some obstacles that she faced when entering into the field of science was really just gender discrimination. Even though she had accomplished a lot, she felt like she was still struggling to break this glass ceiling when it came to gender roles and really just making her mark in a very male dominated field. And because of this, now she really advocates for girls going into STEM and pushing the boundaries for science. Um, like I mentioned earlier, she grew up reading books because she couldn't relate to her fellow classmates and often felt isolated, and she really liked solving puzzles with her family. What I admire most about her is the fact that she empowers female scientists. She was one of the first female scientists to have a really big breakthrough in this recent time, and like I mentioned before, she had some issues being able to make her place and stand her ground in such a male dominated field, but she was really successfully able to do so and helped make one of the most scientific breakthroughs, which is CRISPR Cas9. And her teammate is also a female, and together they won a Nobel Peace Prize in chemistry. She inspires girls all over the world to take on science and make even more discoveries and lets them know that they're capable of creating new discoveries and really making a difference in the world, which is really admiring being a female myself, also planning on going into a STEM dominate or male dominated STEM field. What is she involved in? So basically, her main goals are biochemistry and genetics. Like mentioned before, her biggest accomplishment is helping develop CRISPR Cas9 gene editing technology. She figured out that Cas9 uses base pairing to locate and splice target DNAs when mixed within a guide RNA. And if these cells have CRISPR system encoded in their genome, then a cell is able to make an RNA copy of the CRISPR locus. Basically, this whole process allows gene editing to occur, which is really helpful for modern technology because we can really combat a lot of issues in many different things like agriculture, medicine. CRISPR really plays a huge role in the future of these areas. So, her most recent paper focuses on the successes and current limitations of CRISPR Cas9. This was published actually last month, I think January 23rd, 2023. So, it's pretty recent. And it also talks about the advances that CRISPR Cas9 should be making. And it highlights specific examples in agriculture and in medicine where CRISPR Cas9 is making a really big difference. This figure right here represents how well CRISPR-Cas9 works in different organisms. So the A area is the CRISPR-induced gene knockouts in eukaryotic cells that result from the DNA DSB, which is created by Cas9 RNP, and then commonly repaired and joining their repair pathways. The B talks about the speed of creating knockout mice and other animal models. This is really increased with the development of CRISPR-Cas9 editing technology, which can edit single-cell embryos to create gene-modified mice, which is really, really important to show like just the development of genes. And then the C area right there shows that CRISPR screens are utilized for functional genetic screening, and these Cas9 editors introduce genetic permutations into a model, which is subjected to a selection assay, which is followed by a readout to evaluate the effects of these permutations. So you can see that right here, the modified cells, and then they analyze from selected population with the desired trait, and then identify the RNA sequence present, and then identify the gene sequence of like interest. So basically finding out that particular gene that they want to edit. And then the D section, it represents the fact that CRISPR-Cas9 is a platform for multiplex editing in plants as well. Multiple GNRAs can be used with Cas9 to simultaneously edit multiple targets in a genome, which is shown right there. So it's like you can edit multiple targets in a single genome. And then E shows the site-specific modifications using base editing. And CRISPR-based editors, typically composed of Ncas9 or Dcas9, are fused to enable site-specific modifications without DSBs. So basically, that is just showing how CRISPR-Cas9 can work in like different organisms and in different ways. Next steps, she is very, very passionate about gene editing and taking CRISPR-Cas9 to the next level to make sure that it's being used in different things like the FDA approval of sickle cell therapy, increased nutritional value of more foods, improved in vivo delivery. These are just some examples of ways that CRISPR-Cas9 can really enhance the modern world. And it's just trying to make the world a more efficient and effective place.